Okay, thank you very much. This is my second Li-Fraumeni meeting, and the first one was a real eye-opener for me. It really had a huge impact in my feeling about the work we do in our lab, and in fact, as I will hopefully get to by the end of this talk, I met a Li-Fraumeni family member at the last meeting, and I ended up working on his mutation. But that's at the very end of this talk. So I tried to pose the questions that we would like to address. Whoops, how do I have to learn how to do this? Is it this thing? It's this thing? It's this? Oh, the bars, these. That's why everybody's hunched over the computer. Okay, so let me go back. Yeah, so I tried to pose the questions that I have not yet answered at all, but we would like to at least think in our work that someday the experiments we do might lead to some new ideas about treatment of cancer, particularly in Li-Fraumeni patients. And first of all, I asked David actually what I should talk about, and he encouraged me to talk about some work we'd found with statins. These are the drugs that treat hypercholesterolemia. But I also wanted to talk about some new data that leads into this finding with our Li-Fraumeni person. Okay, so we wanted to ask about statins. We've learned something interesting, I think, about MDM2's ability to degrade P53, which we want to do based on Gigi's findings, that it's definitely important in keeping mutant P53 low in tumors. And finally, something a little bit about what we've learned from our patient. So first of all, we know, thanks to many people's work now, that P53 doesn't regulate only apoptosis and senescence and DNA repair and cell death, but it also has a big impact on metabolism in cells. And people have looked a lot on glycolysis and the citric acid cycle, but there's also another very important aspect of metabolism, which is lipids. And so what we discovered, actually the work of a fantastic MD PhD student in the lab several years ago, was that when we worked with breast cancer cells that only had mutant P53 in them, if you knock down mutant P53, they underwent a morphological reversion to much more normal-looking cells when their cells were placed in 3D culture. And as a result of that, we did gene expression array analysis, collaborating with Arnie Levine and others, and Scott Lowe, I should say, because we all have a grant together. And the pathway that came up as being very significant in the ability of mutant P53 to maintain the malignant characteristics of these cells was the cholesterol biosynthesis pathway. So this is called the mevalonic acid pathway. It's the pathway by which cells not only synthesize cholesterol, but many very key byproducts that play very important roles in cancer-relevant processes, such as prenylation, which anchors RAS and ROA to membranes, and others as well. And they are also, by the way, the targets of the blockbuster statins that many people, many people, including me, take as to maintain their cholesterol levels low. So it occurred to Will to maybe test whether statins might have an impact on the ability of these breast cancer cell lines grown in 3D culture. And indeed, he found that treating the cells with, okay, this works, yes. With basically very low levels, pharmacologically relevant levels of statins, such as simvastatin, actually reduce significantly their growth and malignant characteristics in 3D culture setting. And this was reversed by providing the cells with mevalonic acid, which is basically is what's shut down when you treat the cells with statins. Furthermore, this ability of mutant P53 knockdown to make the cells look more normal was completely reversed when the cells were provided with mevalonic acid, which implicated indeed this cholesterol pathway in the ability of mutant P53 to maintain these cells looking and acting very malignant in 3D cultures. So we also collaborated with Annalisa Borison Dale in Norway, who had, at the time, it was before the TCGA had really become a very useful data set. She had many breast cancer data sets, and she clustered them for us, and we discovered that patients with the highest level of these cholesterol enzymes had the highest level of P53 mutation, and indeed there was even a survival component. Those patients had worse survival that had these high levels of these enzymes. So that work was done, and it led to us asking a more recent question, which is also relevant in the sense that if you have a mutation in P53, you're not only gaining function, you're also losing wild-type function. So the question, therefore, is wild-type P53 actually playing a reciprocal opposite role in the mevalonic pathway? This is not a particularly novel idea. It's really very interesting that many genes and pathways that are upregulated by mutant P53 tend to be very low in wild-type P53 and vice versa. So we asked whether wild-type P53 might repress the expression of the mevalonic pathway genes, and if we didn't have that answer, I wouldn't be telling you this. There was work of Sungwon Moon and a great collaboration with Scott Lowe's laboratory that's ongoing. And what Sungwon did was to show that when you activate P53 with nutlin, which is the way to activate P53, you might say in the cleanest way, because it basically only hits P53 and doesn't do other things in the cell, you see less expression of several of these enzymes. These are liver cells, and these are the famous HCT cells where P53 has been deleted. You see a much bigger impact on knocking down these genes in the presence of wild-type P53. So the question then was, how does this work? And it turns out that the mevalonic pathway is regulated by a master transcription factor called SREBP2. And SREBP2 is normally held as an inactive precursor in the ER. When the cell is deprived of cholesterol, it is then chaperoned to the Golgi, where it is subjected to a couple of cleavages, and then the cleaved protein, the mature SREBP2, goes into the nucleus and activates all the genes in this pathway, or nearly all of them. And so one thing we considered, because all the genes that we tested appeared to be affected by P53, was that perhaps P53 is working somehow to prevent this process. And that's exactly what we found. Comparing, this is just one example of this, comparing wild type to null cells, there was much higher levels of the mature precursor in the null cells than in the wild type cells. So we then, Sungwon spent a year at least trying to figure out what was happening. He tested all of the genes in this pathway that he knew, and nothing seemed to be giving him anything, until he started reading more deeply, and he discovered a candidate, which is one of the transporters that actually is most well studied for being able to export cholesterol from cells. But another group, a group in Japan had published a paper that under certain conditions, ABCA1 can actually retrogradely transport cholesterol back into the ER. When the ER is saturated with cholesterol, this shuts down the cleavage process. And that's exactly what we found, in the sense that in the cells we've worked with, if you knock down ABCA1, you see significantly more mature SREBP2. So this then led to the possibility, well, is ABCA1 actually a target of P53? And it turns out it is. If you turn on P53 with nutlin, you turn on ABCA1 message and protein. We've shown that in several cell lines, and in fact, we have cell lines where we can turn on wild-type P53 without any kind of a stressor, or a P53 that's transcriptionally inert, and that will shows that only the wild-type p53 can actually turn on ABCA1. So we believe that at least one of the ways in which p53 is shutting down the pathway is by activating ABCA1. So what does this have to do with a more physiological situation? First of all, we started collaborating with Scott Lowe, and Chun-Hao Wang in his lab did an analysis of over nearly 500 different cancer cell lines. Those with wild-type p53 had very low levels of mevalonic pathway gene expression, whereas those without p53 or mutants had much higher levels. This then led him and eventually another collaborator to do some experiments in mice, which I think were very confirmatory and extended what Sung-Wan had found. This is a volcano plot, I think that's what it's called, where basically an older p53 model had been used in Scott Lowe's lab, where you can actually create tumors in mice that lack p53, then reactivate p53. And when p53 is reactivated and the gene expression is analyzed, what they found was that there was a big drop in many of these different mevalonic pathway genes and a corresponding increase in ABCA1. But more recently, a more somewhat high-tech model has been used in Scott Lowe's lab, where they inject into the tail vein of mice constructs that are CRISPR-based that will either knock out p53 or other tumor suppressors, such as axin, which is in the Wnt signaling pathway, along with MYC, which is a well-known oncogene. And these mice get hepatocellular carcinomas within 4 to 6 weeks. It's a very fast process. As Scott has mentioned, previously it would take a year or two to be able to develop these tumors. And so one of the things we discovered was that the p53 null tumors have much higher levels of mature SREBP2 than the other tumor suppressor-derived tumors. And so that led Chun-Hao to think, well, is this pathway, this mevalonic pathway, actually something that's needed by these tumors to form? And so he knocked down 3 different genes in this pathway by combining the knockout of p53 plus MYC plus shRNAs driving various genes in the pathway. And this is just to show the process, how we did it. And quite extraordinarily, there were virtually no tumors in these mice when he ablated these tumors in the mevalonic, when he ablated these genes in the mevalonic pathway. And by contrast, in the axin tumors, it didn't have any impact on their development, or had much less impact. So this led us to think, well, maybe you're not obviously going to treat a patient with shRNAs against various genes, but statins might well be an effective prevention in this case. And so when they pre-treated the mice with the p53 mice with statins, there was a significant drop a significant decrease in their tumors, the number of tumors, as well as the volume, the weight of the livers with the tumors in them. In contrast, there was virtually no impact of statins in the tumors that were driven by loss of axin. So that is basically our data that suggests that statins, and we think it's prevention, by the way, because the mice are pre-treated with statins before they're given these various constructs. And at least in a preliminary experiment, they didn't have as dramatic an effect if once the tumors were formed, the mice were then given statins. And just some new data here. As I said, ABCA1 appears to be at least a contributor to the tumor suppression by p53, but this is p53 survival curve. This is the survival numbers with knocking out ABCA1, and you can see it's definitely a tumor suppressor in the sense that the mice are getting tumors, but they're later and fewer than the case with wild-type p53, which is totally consistent with the fact that p53 has multiple targets in cells, and probably many of these are contributing to its tumor suppression function. But interestingly, you do see in cells from these tumors much higher levels of SREBP2 mature form. So this is the model, and before I get on to the other part of the talk, I haven't even had the courage to look at my watch. But basically, a wild-type p53 can basically activate this pathway, and this should say ABCA1, it's an old slide, whereas mutant p53, which I didn't mention, actually helps SREBB2 activate the mevalonic pathway by a mechanism we don't fully understand. So I'm going to now switch gears and talk about newer data that was just published, and then that will lead me into some very new data. This is the work of Hen Cass and several collaborators. And what Hen had discovered was that if you turn p53 is normally found as a tetramer, 4 copies of p53. If you force p53 to become a dimer, it becomes a much better target for MDM2 degradation. This is just a few examples here. It's a very busy slide, but you can see in almost every case, except when p53 is a monomer, it's much better degraded. And you can see this better when you look at the half-life of the protein. The half-life of the protein is always much shorter, essentially, when p53 is a dimer in the presence of MDM2. MDM2 preferentially degrades dimeric forms of p53. We did a lot of work. We know how p53 is interacting with MDM2. It's a new binding interaction. And we also know that the dimer form is actually, degraded by the 20S proteasome, not the 26S proteasome, and it's a ubiquitin independent manner. And we've even shown that wild-type p53, the dimer form of wild-type p53 is degraded by the 20S proteasome and not by the 26S proteasome. I can tell people the experiment in more detail if they're interested later. So, there is some potential relationship between mutant p53 and MDM2 as well when it's a dimer. So the idea was, if we were to force a p53 mutation, such as we find in many human cancers, including Li-Fraumeni syndrome, into a dimer form, would it be better degraded by MDM2? And the answer is yes, in some but not all cases. In some cases, we see a significant increase in degradation, and others we don't. But in those that we do, we can even, this is, Hen loves to put in little cute signs, symbols there. But what we did find is actually in a very simple assay where you basically scratch away cells in a plate and then measure their ability to basically grow into the cell. This is called a wound closure assay. And these mutant forms of p53 do it very well. In the presence of, or absence of MDM2, except when the p53 is a dimer, and then it's much less effective at this wound closing activity. So the ability of, which is consistent with the fact that MDM2 is probably degrading that mutant p53. So that's basically where we were. We published this paper. And then when I was at the Li-Fraumeni meeting, I guess it was 2 years ago in Columbus, Ohio, I happened to meet a member of a Li-Fraumeni family, and we got to talking. Very nice man. He's not here. And he contacted me afterwards and said, "Can I help?" And so I contacted our collaborator, Paul Huang, who's here, and put him in touch with this patient, and he gave some of his fibroblasts to them, and we've been working with it. And he is consistent with what I've just been telling you. His mutation is actually in the oligomerization domain. And this is actually taken from the IARC database. I think it was done by a fantastic undergraduate in the lab who's been working on this project, and I hope it's right. About half of these mutations that are in the oligomerization domain are the mutation, or mostly the mutation that Jerry talked about, but there's a couple of others as well. And in fact, A347D is the third most common mutation within the oligomerization domain. It's still a small number, I have to say, but there are families or patients with this mutation. And so oh, I'm almost finished. You'll be happy to know. So maybe not. I hope you won't be that happy. So P53, basically, we've done some experiments with this patient's fibroblasts. And here's the problem. I mean, we clearly see, I hate the word clearly because you always feel defensive, but we do see that there is definitely more P53 than when we compare this patient's fibroblasts to other fibroblast cell lines. The real problem is, and this is something we have to deal with because we want to be working with cells from patients, is we really need good control cells. And so, in fact, I hope that I can have conversations with both scientists and doctors here to figure out how we can increase the bank of controlled fibroblasts we have. At least with all the fibroblast cell lines we've looked at, there's always more P53 in this patient's fibroblasts and correspondingly much less MDM2. But to at least get around to, and I should say that when we've done a transcriptional analysis, although there are some interesting exceptions, the transcriptional activity, these are the orange bars, is always somewhat less than, or sometimes a lot less than the control cells that we've looked at. So in order to at least deal with controls and to understand in a little cleaner way, we also generated CRISPR-derived U2OS. These are osteosarcoma cell lines. And we generated a number of clones. These were done by a fantastic former lab member who's now an MD PhD student. And basically, he knocked into U2OS cells he generated either HETs, would be one wild type and one mutant allele, or what we call HOMs, would be two mutant alleles. And we did glutaraldehyde cross-linking. The vast majority of the P53 protein in these cells, in the HOMs, is a dimer. You see more or less an equivalent distribution in the HETs, and then although this is very faint, there's practically no detectable dimers in the wild-type clones. When he actually looked at the levels of P53 in these cells, the HOMs have rock-stable P53. This is kind of analogous to what David was talking about this morning, in the sense that you cannot get any higher levels of it once you treat the cells with P53 activating nutlin. And that P53 appears to be very weak as a transcription factor. In fact, there's a very nice series here where you get higher levels in the wild-type clones, intermediate levels in the heterozygous clones, and very weak activity in the homozygous clones. Now we've just started a program, and this is the last slide I'm going to show, to see whether these cells show differential responses to different kinds of chemotherapeutic agents. And one potentially very interesting result we have is that the HOMs, the ones with only the mutant in it, are much more sensitive, actually, to etoposide than the wild type. So we don't understand why this is, but it is, and we hope to extend this and understand more about different sensitivities of these patient cells to different chemotherapeutic agents. So these are our goals based on what I've told you. We're hoping to convert mutant P53 to dimers in mutant P53 cells. This is kind of a long-term sort of dream type experiment where we have all kinds of ideas how we want to approach it. We want to test, we want to take the Li-Fraumeni-derived fibroblasts and engineered tumor cells and test both pathway-derived chemotherapeutics and maybe statins as well to see if we can see differential sensitivity. And finally, we want to expand to other Li-Fraumeni mutations in the oligomerization domain. So that's I'm finished. And this is my lab. Sung Wan did all the work on statins and wild type P53. David Tong, who's now in medical school, made all these CRISPR-derived cells. Hen did all the work on dimer-forming mutants, and Josh is now working on these Li-Fraumeni patients-derived U2OS cells. Thank you.
Thank you for your talk. I wonder with the statin data, has anyone looked at the converse? In other words, do patients with high cholesterol do they have increased tumor burdens or increased risk of tumors? Is there some relationship between cholesterol levels and tumor onset?
I actually think there is. I don't know anything about that in Li-Fraumeni patients. What I do know is that there have been a lot of meta-analyses about statins, and it seems to be truly protective for liver cancer. Now, liver cancer happens to be rather rare, as I understand, in Li-Fraumeni patients, but breast cancer is another one that I think is really worth looking at.
So first of all, it's really a great talk, but I just want to say that at that dinner in the evening, where you talked with the patient, I witnessed your conversation, and I was thinking, how great are these special meetings where they...
Absolutely. Yes. This guy changed the direction of our research.
Inspired us, basic researchers, to do something practical.
He said, can I help? And, and then I have to thank Paul Wong, who's here, because he was really incredibly helpful in this. Thank you.
So all the people who are doing this effort to bring the scientists in your form of patients and doctors are really doing a very important mission. I think the best mission in P53.
Thank you.
I already had my question. Okay, great. Any other further questions? Yes.
So I was wondering if you looked at knocking down LDL receptors. That's another important SRBP target to get a mechanism.
Yeah, yes, we're just starting to look at that, to be honest. And I just want to say generally, lipids are like sort of the wave of the future in terms of cancer biology. I keep hearing interesting things about lipids.
So I'll do a wild card.
Yes.
How are you thinking of making them dimers, mutant P53?
Okay, so yes.
What's your favorite idea?
My favorite idea is to capture Brent Stockwell and get him to help me screen. We figured out all kinds of probably wacky assays we've thought of, like sort of with, what's the word, ligation-mediated, fluorescence assay, to do that.
And then just to screen.
Here's something very interesting. Well, it's not that interesting, but dimers, I think it's very interesting, but I might be the only. Dimers form co-translationally, tetramers form post-trans, and we've actually confirmed that. It's very interesting. Yeah. So you can do it.
All right. Thank you very much.